So a lot of times women are being tested for mutations in BRCA1 because they have a family history of breast or ovarian cancer. So to be told that they have a variant, but the doctor doesn't know what it means, does not reduce their stress or reduce their anxiety. We're trying to give the clinicians information that they need to understand whether or not a variant would increase cancer risk or be perfectly harmless.
An MD-PhD student in our lab, Greg Finley, developed a technique called saturation genome editing, in which we basically are making every possible change in the gene BRCA1 and then classifying each possible change into the ones that are disease-causing and the ones that are perfectly harmless. So the way that we did this with CRISPR allows us to classify these variants with high confidence into either being disease-causing or being perfectly benign. And that information, essentially when passed on to women, allows them to have confidence that rather than uncertainty about the information that they're getting and how it relates to their health. So our plan is to extend this approach to additional cancer risk genes that are known over the next year or two, hopefully covering them all. So one of the exciting things that we found in this study is that you can study changes in the BRCA1 gene in the lab, and they actually reflect with surprisingly high accuracy what happens in a person. The premise of genetic medicine is that what we've been promising for now 10 or 15 years is that we'll sequence the genomes of ordinary citizens, and that information will somehow allow us to improve how they are treated, how their health is managed. And it's been challenging to deliver on that promise for a few reasons, but one of the key reasons has been this challenge of interpretation. We can now make interpretations with confidence. And this is just one gene, but if we can take the same paradigm and extend it out to other genes, then maybe we're on the way home. Our hope is that this database, and the database will also continue to grow, will become a central point for essentially guiding the interpretation of these, quote, actionable genetic variants as they are first observed in women, thereby just making it possible to actually deliver on the promise of genetic medicine.